All right. Good evening, everybody. Thanks so much for joining us this evening. My name is Eric Austin. I am the director of the Wheeler Center. I am also a member of the Political Science Department here at the university. It is a great honor to welcome all of you. Um, I'm always a little sort of a combination of nervous and excited to make these kind of welcoming remarks at our events. Um, Partly because despite having spent, I don't know, 20 odd years in this career at this point, I still get stage fright when I get up in front of a group. But I'm always excited because it means that we have sort of gotten through all of the hard work of preparing for an event like this, and we get to really sort of sit back and enjoy the fruits of the labor. That's even more true, I think, for this event than many of the ones that we've hosted in the past. Like a lot of organizations, the Wheeler Center has had to sort of put a pause on a lot of its programming activities and has had to become a little more creative in how we produce events, given everything that's happened in the last 12 months or so. So the fact that we're able to host this event this evening is particularly gratifying. These events, the politics and pizza events, that this is part of our larger series. Were really the brainchild of one of my former colleagues, Dr. Linda Young, a number of years ago when she was the head of the Political Science Department. And she was really looking for an opportunity to engage students in conversations with the faculty, both in the department and across the campus, as well as other experts around the community on topics that were more timely and were emerging quickly or were of interest to students in other kinds of ways that maybe we couldn't get to in our regular courses. And when I joined the Wheeler Center a number of years ago, it was pretty clear to me that these politics and pizza events were also a really great fit with the mission of the Wheeler Center to engage and address issues and problems that were critical to the citizens of Montana. And so being able to partner with the department and the Wheeler Center was kind of a no-brainer in a lot of ways. Um, these particular events, I think, also really capitalized on and sort of recognized the vision of one of our longtime board members at the Wheeler Center, who really wanted to create opportunities for the Wheeler Center to engage with students at the university as future problem solvers and policymakers, and honestly, in a lot of cases, current problem solvers and policymakers. That board member was also, I think, really a critical and sort of instrumental individual in bringing a vision to the university that moved MSU from a storied but relatively small sort of statewide or regional institution to the world-class university that it is now. And that board member is the former president of the university, Bill Teets. As many of you know, Bill passed away about a year ago, or in the course of the last year or so. And I wanted to sort of acknowledge, given that this is the first event that we've had since his passing, wanted to acknowledge and kind of recognize the contribution that Bill made both to the Wheeler Center and to the university as a whole. He will be greatly missed. Um, Having said that, though, I want to sort of turn to a slightly happier note and take a moment to recognize the hard work of two individuals who really drove tonight's event forward. And that is Carolyn Rader, who is the Wheeler Center's fellow this year, and Melissa Wheatus, who is our intern this semester. When I talked to them early on about sort of creating this event or taking on this event, they jumped both feet, sort of full force into figuring out how to make this work in a very different environment. And they have been extraordinarily creative and innovative in figuring out how to make this work. And so seeing 40 odd participants listed in my WebEx screen, I am really, really grateful for all of the efforts that they put together. So please, either virtually or if you can turn your screens on, please join me in giving them a quick round of applause. So I also want to briefly acknowledge the board of the Wheeler Center. unfortunately, our board is not only spread out all around Montana, but around the country as a whole. And so it is often difficult for them to join us for events that we hold on campus. It's a pretty long trip to get here often. With a virtual event, we're able to do this in kind of a new way. So I'm really grateful for their ongoing support for these events, as well as the leadership and the direction that they provide to the Wheeler Center as a whole. Having said all of that, I want to get out of the way. You didn't come to listen to me, and so I am very happy to turn the microphone over to Melissa to introduce our panel for tonight. Before I do that, I almost forgot. I have one quick announcement to make. And that is that if you are interested in this particular topic of the COVID-19 virus and emerging vaccines and vaccine protocols, I want to encourage you to be aware of an event that is being hosted by the Center for Science, Technology, Ethics, and Society. They will be hosting a WebEx event on April 6th at 6 p.m. and we'll continue talking about many of the sort of related issues to the ones that we'll be exploring tonight. So please keep that on your calendar and on your radar. And with that, Mel, take it away.
Excellent. Well, once again, thank you very much, everybody, for attending this evening. After our four panelists have given their brief talks, we will be doing a Q&A session in which you are all able to ask any questions you might have. I'll be explaining that more a little bit later, but I am thrilled to announce our four incredible speakers lined up for you this evening. Our first speaker of the night, Dr. Matt Taylor, is an associate professor in microbiology, immunology, and biotechnology, specializing in pathogen evolution. Our second speaker of the night, Dr. Seth Walk, is an associate professor in microbiology and immunology, whose lab specializes in infectious diseases as well as microbial ecology and evolution. Our third speaker is Dr. Liz Shanahan, a professor of public policy in the Political Sciences Department, who specializes in policy narratives. And to round out our speakers, we have Mr. Christopher Coburn, a member of Gallatin County's Board of Health and Vaccine Distribution Task Force, here with us this evening. Without further explanation, let's get started with Dr. Matt Taylor.
There we go. Hello, everybody. I'm Matt Taylor. I'm in the Department of Microbiology and Immunology. I'm a virologist. I used to study herpes viruses until last year. I jumped into driving some of our live virus research, looking at SARS coronavirus infection in cells. So the pandemic has been going on for over a year now, first starting a little before Christmas of 2019 in Wuhan, China, and rapidly spreading across all 7 continents. Antarctica succumbed to COVID infection just before Christmas Eve of 2020. There have been 111 million people infected worldwide, almost 2.5 million deaths. Domestically, we have hit a landmark of having over 500,000 people in the United States succumb to COVID infection. Case fatality rate has varied from between 0.03 to 0.5%. I don't think I really need to stress the fact that this is a very serious problem that has changed the way we all approach our life. And it's all caused by this little bug here. It's a virus known as a coronavirus, SARS coronavirus number 2. These are so small we can't see them by the naked eye or with a microscope. We have to go down to electrons to visualize these viral particles that transmit from person to person, causing this disease. On the right is just an image of what that electron micrograph looks like. It's a spherical virion. This is that, again, that infectious particle surrounded by this crown of what's been figured out to be the protein. This is the spike protein. And the spike protein is going to come up a number of times because it's incredibly important, not only for how this virus transmits and infects cells, but also how we have developed our strategy to control it with vaccines. This is a schematic of that virion on the right. It is an RNA virus. Its genome is packed inside, tightly wound with this nucleocapsid protein in a helical symmetry. It's wrapped in a lipid envelope, which means it's really susceptible to soap. So if you take away one thing, stop touching your face and wash your hands, because it's a really effective way to combat these envelope viruses. I also want you to pay attention to these orange spike proteins. These are the proteins that mediate entry of the virus into the cell. They're also the target that are being used in that vaccine strategy. Immune responses to this spike are amazingly effective at blocking infection. Now, SARS coronavirus causes this umbrella of diseases we've termed COVID-19. It gets its name, severe acute respiratory syndrome virus, for the significant lung pathology. There's a lot of inflammatory infiltrate, cytokines, and immune responses that compromise lung function, causing people to have trouble breathing and sometimes a lethal illness. But that's not to say it doesn't infect other tissues. In fact, we found SARS in the brain, the kidneys, the liver, the intestines, and the heart. And all these tissues can suffer from damage of SARS coronavirus infection, resulting in a very wide range of illnesses. Some that are acute and self-resolving, some that last for extraordinarily long periods of time, where these people cannot seem to clear either the infection or the associated illness, this so-called long COVID. What we have learned, though, from people that have survived from COVID infection is the kinds of immune responses that are protective. And one of the critical features, not the only feature, but one of the major ones that is focused on, is the production of antibodies that recognize the spike protein. So as I mentioned, that virion, when it needs to enter the cell, needs to bind to a cellular receptor known as ACE2, at which point the spike facilitates that entry of the virion, initiation of infection. Antibodies that bind to the spike prevent that activity from occurring, neutralizing the virion and interfering with the process of infection. We've used this for some therapies, such as the Regeneron and other monoclonal antibody cocktails that have been given to severely ill individuals, but it's also the goal of most vaccines to derive an immune response that is going to protect against infection. Besides that, this pandemic's been going on for a year, the rate of vaccine development has been incredible and unprecedented. As early as 2 months into the pandemic, we already had vaccines in development that have already now received approval, faster than any other approval that has been seen to date for an infectious disease, but only because they built upon technologies that have been in development for decades. The leading candidates are part of these 4 approved vaccines, which comprise 2 different strategies: a nucleic acid or mRNA vaccine, this is the Pfizer and Moderna, as well as these recombinant viral vectors that use replication-incompetent adenoviruses. That is not to say that all vaccines have been successful. We've had a couple that have been abandoned, but that is not to mean that they were flawed. It just means they weren't as good as some of the other vaccines that were in development. Just because this is fast, just because it is happening at an unprecedented scale, does not mean that these vaccines aren't any less effective. In terms of the strategy for these vaccines, they both are working on the same principle, which is to deliver a nucleic acid that will result in the expression of that spike protein by your cells to be recognized and cleared by your immune system. The Pfizer and Moderna vaccines are these mRNA expression vectors. They are RNA that's packaged inside a liposome, think of it as like a balloon that gains access to the cell, delivering this message that is going to get translated to make the proteins. So your own cells are making these proteins. There's no risk of an infectious virus being produced from an mRNA mechanism. It's an extraordinarily safe system that has been, again, developed for a long period of time based on a large amount of very sound research. It has resulted in a highly efficacious response. Over 90% of people are protected from hospitalization or death from COVID-19. They elicit strong antibody responses. The limitation is that they require extraordinarily cold storage conditions. Now, the other major strategy that has been receiving recent approval are these adenovirus-based vectors. This is the Oxford AstraZeneca vaccine, the Sputnik vaccine out of Russia, and the new Johnson & Johnson vaccine. These are non-replicating viruses that do the same thing. They deliver a nucleic acid to result in the expression of that spike protein in your own cells to be presented and cleared by the immune system. These are also very efficacious vaccines. They are reducing hospitalizations and they're preventing infection. They are also having the benefit that Johnson & Johnson vaccine is showing great efficacy towards a single dose delivery. Not only that, but they are simpler to store and they have better stability than these mRNA vectors. And so it comes to this contrast of the benefits of different strategies towards creating a most efficacious vaccine, which is a measure of how effective that vaccine is. We have to consider it from multiple levels. Is it safe? Is it effective? And is it practical? Safe means that it has to have minimal side effects. It cannot be worse than the disease we're trying to prevent. In fact, all of these vaccines have shown Minimal side effects, though people are worried. I will tell you from personal experience, it is no worse than a tetanus shot. If you've ever had a tetanus shot, you know your arm's sore, but it's better than getting tetanus. They're also very effective. They elicit good, strong protective responses that prevent illness. We can measure that, like this study here from the Moderna vaccine that compared a group of placebo versus people that were given that mRNA vaccine. These are people who have been diagnosed with COVID-like illness or COVID illness, and showing that these people that have received vaccine are at a significantly reduced risk of developing any amount of disease. That's where your 95% efficacy comes from. We also have to consider that practicality. Are they stable? Are they easy to administer? And they're not cost prohibitive. All of these measures are going into that idea that we are trying to convince the public that there is a greater benefit to receiving the vaccine than the risk to getting the illness in the first place. But vaccines are just but one part of our public health policy to prevent the spread of infection. This is a great model that came out about 6 months ago. It's known as a Swiss cheese model, which considers that there's no perfect prevention to get to blocking the transmission of COVID in our communities. But by using a series of good, not great, but good measures, we can significantly impair the spread of the virus from person to person. This includes the use of masks, staying home when you're sick, washing your hands, not touching your face, and vaccines are that last measure. We can control the spread in our communities if we use all of these judiciously. And I think that's my 8 minutes, and I'll stop talking.
Thanks, Matt. That was great. I'm going to switch gears a little bit and tell you how we detect the virus in some of our testing activities right here at Montana State. And I'm going to pick up there on that spike protein that Matt spoke about earlier. And in this Scooby-Doo like cartoon here, these green proteins protruding from this virus, you can see that interacting with the host cell. And that spike protein is why the vaccine targets, but it's also important for entry into the cell. And I'm just wanted to remind myself, if I can advance my slide here. There we go. Remember, coronaviruses came, or at least have gone between humans and animals for quite some time. And so this one came out of the blue at us because it's probably the first introduction into humans, at least on this scale. So it was really new, it was novel, and it took us by surprise a bit and got us into a tight spot. And around about July, the state reached out to MSU to see whether we could increase testing capacity throughout our region. And we were happy to help that cause. We put together a team pretty quick and were able to get to work with the CDC protocols on establishing testing standards right here on campus. Now, the CDC originally brought this test out that looked for the genome of the virus. And this technique that we use is called PCR, or quantitative PCR to be more precise, to actually detect this virus. If you want to know more about that, I put a link here. But the CDC really is the governmental agency that drives pandemic responses in our nation. And so this development of this test was not done lightly. It was very rigorously made, but it was very quickly implemented around the country. And so we got a hold of that protocol and began doing that here. Now, I already mentioned this, but I just want to reiterate. This test looks for the virus genome. It doesn't look necessarily for live viruses. And because of that, we don't really know if everywhere that we detect it, it's actually going to be infectious. And that's good somewhat, because we want to err on the side of caution. We want to know whether it's there or the presence of the virus is there. And that's going to help us create strategies like public health rules that keep people safe. And I do also want to mention that this is the gold standard way to diagnose this disease, is by looking for the virus with this technique. There was a lot of, I don't know if everybody remembers, but back when you went in for a COVID test, say over the summer, there was this really painful procedure where they would basically shove a swab up your nose and get way back in the back and cause you to sneeze and gag and everything else. And that was problematic, not only because it was invasive and it kind of hurt, but caused people to sneeze. And we learned very quickly that this thing is airborne and that's how it gets around. So it causes a lot of issues in that. We still use nasal swabs, but we don't go all the way back anymore. We tend to just get the middle part of the nose, and that's proven to be pretty effective. So qPCR, looking for the viral genome, that's the gold standard. Now, you've also might have heard about these antigen tests. Antigen testing, or antigens, refer to a foreign substance in the body, so something that's not normally there, and it's our immune systems go and surveil for these things. They look for them and they clear them out. And we can capitalize on that biology by making molecules that recognize antigens. We actually have cells in our bodies called B cells or plasma cells that generate a lot of antibodies, and they're very specific when they do that. So if we know what we're looking for, we can generate these antibodies to look for them. And there's companies out there that began making antigen tests and rapidly deploying those out. And there's more information about antigen testing on this website at CDC. I did want to put this table up just to illustrate a few points. Although qPCR, like I said, is the gold standard, antigen testing became very popular. And there's good reason for that. It's generally less expensive, but the turnaround time to get the result is much shorter, okay? The problem is that it's not quite as accurate. And so there's a balance here. There's a trade-off. And as Matt was talking about, through this pandemic, we've learned a lot about trade-offs and risks and how to weigh risks and what factors are more important. And the interesting thing about our society is, depending on which community you live in and which societal factors you value, that influences a lot of how you weigh risk. And so while some parts of the country, like ours, were just fine doing the RT-qPCR testing, a lot of other places around the country switched over to the antigen-based testing. And they had reasons for that. And those reasons weren't necessarily right or wrong. They were just the values of that community. So I did want to talk a little bit about logistics. So one of the other things I would love for everyone to consider is, why not test everybody all the time, everywhere, until we can figure this thing out? And that just has to do, I think, with logistics and how we get things done in a way that meets federal guidelines for diagnostic testing and gives people the right result. And that all starts with our public health system. And in Montana, we have the Department of Public Health and Human Services that's up in Helena. And they kind of get their marching orders from the governor's office and our state government. But they're also taking guidance from the CDC and the federal guidelines. Okay, so these are the folks that try to coordinate vaccine responses, diagnostic responses, responses to any infectious disease in a very concerted way that is consistent with the way that the rest of the nation deals with these things. Now, we get our samples here at MSU from DPHHS, we test them, and we send back their results. At that point, the results get entered into our state database, and then those results are reported to local departments of health. At that point, contact tracing is implemented, so our public health practitioners can go out and try to identify outbreaks or folks at high risk of spreading the disease and do certain protective measures like isolation and quarantine. Okay, so there's a lot of logistics here. By the time we get the sample, it takes us, well, at the peak of our production, when we were doing about 2,000 a day, it takes 24 hours at least to return results back. So that's even after those were collected and had to be sent to Helena, had to be sent here. And then after we send our results up, it takes a while to process and get things through the database and get the results reported in a consistent way. And so the time between when you get sampled and your test is delivered, your test result is delivered to the local public health, is at least 2-3 days. And it's really hard to cut that down anymore just on our infrastructure. The other thing I wanted to talk about, these are some pictures from our lab. These pictures are just meant to illustrate that there's a lot of stuff that we need. We need a lot of equipment, and we need a lot of supplies and reagents to run our tests. And nationally, through this pandemic, there was a shortage of almost everything you can imagine. There were shortages all the way along our production line, the biggest one being this right here. These are called pipette tips, and these carry the sample from a test tube into our testing platform, into our instruments. And the world has gone through a shortage of these things all over, not just in the US, but all over the place. Another shortage were tubes, and we get tubes of any shape, size. I swear, if you have a tube in your drawers or anything at home that's made of Tupperware, we've probably seen it in our lab. So it's been a real challenge to keep up with these things and to stock just enough tubes to sample everyone over time. I did want to throw this up. I'm pretty proud of this. This is our production at Montana State for our diagnostic testing. On the y-axis here, you see the number of tests that we performed. And then this is on sort of a daily level. Okay, so at one point we were doing up around or just over 2,000 tests a day. And you can see we got pretty consistent numbers every day. But over here you see a bunch of spread of these circles. That just means the number of tests that we do every day is kind of variable, and it's coming down in most cases. That red line is our average, or sorry, yeah, is our weekly average of the number of tests. So we're doing pretty good when we're not having to test a bunch of folks. The bigger news, though, is this positivity rate has plummeted. So of the tests that we get, fewer and fewer of those are positive. And that's what we want to see. And I just have to end here by sharing the names of everybody in the lab that makes this thing work. Just supported by a heck of a team. And these are the most amazing people in my life right now. So thanks.
Hi, Liz Shanahan here. I'm a professor in the Department of Political Science. 50% of my time and 50% of my time I am new in this position as Associate VP for Research Development in the VPR's office. I'm presenting to you some of results of some COVID research. I'm a social scientist, so this is the social science part of the presentation here. And you can see in the bottom of the slide our funding entities. And this is my amazing team, Seth. I pulled pictures, but we're from all over the country, lots of state universities here too. So this is a pretty great team, and it's been really impressive what states have ponied up for funds. So the CDC, Center for Disease Control and Prevention, is the premier science agency in our country, and they recommend getting a vaccine. So this talk tonight is about what that means in terms of the public, but based on these clinical trials, they say you need to get a vaccine. And so why? And I think Matt got to this. You reduce your chances of getting COVID. If you get COVID, you reduce how sick you will get. That this has resulted in a reduction in hospitalizations. And you also simply reduce the amount of viral shedding. And so you protect people around you by getting a vaccine. So how do we think about vaccines and vaccine adoption? You can think about this in terms of this continuum, where there are some people who just simply refuse to get a vaccine, some people who say yes, they vaccinate if the vaccine is available, and there's a swath of people in the middle who hesitate in some capacity. They might lean refusing, they might lean to accept, but they're somehow unsure. And this is the population the CDC focuses on, or the county agencies here, I assume too, and MSU perhaps. This is the population to focus on for risk messaging to nudge people to get the vaccine. So a bulk of my work is trying to understand what kind of risk messaging is effective to move people to get COVID-19 vaccine. So with my team, we have 2 survey efforts largely funded by the National Science Foundation, what's important in these efforts is that I want you to get from this slide is that these are panel surveys. And what that means is that it is the same respondents asked the same questions over time. So we get some longitudinal data. The first survey effort, we surveyed 6 states and we were trying to get representation regionally. The second funded effort, we launched a national survey, and the results here will just be of wave 1 out of that survey effort. All right, so what do you see here? We want to know intent to vaccinate, just sort of broad brush here. Interestingly, in the 6-state survey from May to October, we saw a decrease in people who said, yes, I want to get the vaccine. But then some news started coming out about the Pfizer and Moderna vaccine, and we saw a slight increase of a willingness to get the vaccine. And from the 6-state survey to the national survey, you can't really totally compare one survey to the other, but our results are holding steady. So about 41% saying yes, they're getting the vaccine, 23% saying no, and that important in terms of messaging, middle unsure population at 36%. So we also want to understand variation within why people, say yes and no and not sure. So what are some of the indicators of variation here? And so the first thing we took a look at was ethnicity. And what you hear about a lot in the news and is borne out in our data certainly is that of the people who are saying, yes, I'm going to get a vaccine, very few are Black and multiracial. And the majority of those who say, yes, I'm getting the vaccine are white and Asian. We also looked at partisanship. Again, our data bears what you, I think, have heard probably more colloquially in the media, where those people who say, yes, I'm getting the vaccine tend to be liberal. Those who say, no, I'm not, higher proportion are conservative. And then those who are unsure, it's sort of spread across the continuum of political ideology, but the greatest of that population being the moderates. Now, is there a difference regionally? And again, this is hard to compare because we have a state and versus a region analysis right here. But I just want to focus on one region in particular, and that is the South. In the South, you can see that of those who are willing to get say, yes, I'm getting the COVID-19 vaccine, very few out of the South, in contrast to of those who say no. The South has a strong representation there. And we see in our state data, Michigan and Louisiana are also sort of on that low end of willingness to get vaccinated. In the middle here, this unsure population, we don't have a geographic locale for that. So what that means is that risk messaging really should have a national scope. So additionally, why, gosh, there we go. Why would this, what would actually increase the confidence in COVID-19? So what would make people feel more confident about getting the COVID-19 vaccine? Well, we have some good news here for some of our scientists and in the crowd and those who are training to be scientists, in that even though we have a lot of uncertainty or a lot of talk about how there's a distrust of science, the highest responses really are about FDA approval. Experts I trust say it works and is safe, and healthcare providers recommend the vaccine. So when science comes through, people do listen. The other area that increases confidence is really the human-to-human effects here. So waiting for others to see if it's safe, and then when people I know will get the vaccine. Okay, let's talk about risk messaging. Very briefly, scientists and the public speak different languages. Science talks in the language of probability, frequency, magnitude. And humans, the public, regular people, your mom, your dad, your neighbor, you talk in stories. And so we lean in my work in the narrative policy framework, we lean into our literary scholars and use the word homo narrans. Homo sapien is that we are very analytical and we're knowing beings, but we're also homo narrans in that we are communicate who we are, and we're also shaped by stories. And how the power of story is in what's called narrative transportation. And what that means, and you all have had this experience of going to a movie or reading a good book where you are totally immersed in the story and you have this affective experience that rises and falls with various characters of that story. So the greater the transportation, the greater the power of that story. So what does this have to do with risk messaging? Well, let's take a quick peek at risk messages that we've seen about COVID-19. This is from the CDC. Act now. Maybe this looks familiar from the MSU webpage. I typed those. These risk messages are what I call non-narrative. They are not narrative. These are imperatives of what to do. They are science-based, right? We know that there's science behind masking up that prevents viral transmission. That's all really good. I'm going to make the claim that these are non-narrative and that we can do better. So let's take a look at our experiment. So what we did in our national experiment is that we have these different conditions. So the control condition or the sort of treatment as usual is that imperative, get the vaccine. Well, we kept that call to action. We held that constant across our 3 narrative conditions that you see on the left-hand side of the screen. So what's narrative in this? Well, protect. Part of what makes a narrative is our characters. And the word protect evokes this idea of hero. And so what we do in these messages is situate the target audience as the hero who protects. What we manipulate across these different conditions is the victim. So who is that hero protecting? Yourself, which is the individual, your circle, so people you know, your community, sort of the broader community. And survey recipients received 1 of these 4 conditions. So the analysis I did was a moderated mediation model. So on the left, you'll see the independent variables of treatment conditions, those 4 treatment conditions. The affective response is the narrative transportation. So we want to know what kind of response affectively people have to these different conditions, and if that mediates the motivation to vaccinate. But we also have this belief system. So those 3 narrative conditions, individuals, yourself, circle, and community, are reflective of this American political ideology that we have in this country, that we have individual rights, and think about this on a continuum, to a community interest. But we, as adults, have already these very deeply embedded beliefs about individual liberty and collective community interests. So we want to see if those beliefs moderate any of these relationships. So what you have on the x-axis is that individualism to collectivism worldview. So the standard deviation below the mean is individualism, the zeros are the mean, and the standard deviation above the mean is collectivism. And what you see here, this is valence. So this is looking at that pathway from the conditions to the narrative transportation. And indeed, the control condition tanks, right? So there's very little variation in that affective response. And what does really well is the protect your circle and protect your community. And so that relationship is indeed moderated by this belief system. And we have a significant pathway from affective response to motivation to vaccinate. So we do, in fact, have a mediating relationship here. So looking at the direct relationship, really interesting results also. We have a moderating response whereby the collectivism worldview does correspond with a stronger motivation to vaccinate. And again, we see with these conditions, the protect your community, protect your circle being the top 2, protect yourself, being a little bit below that in terms of motivation to get yourself vaccinated. So what do we have here? What's the point? On the top, we have our Bobcat risk messages. And my point is we can do better, and we should do better at nudging, at motivating, and can do better through narrative. And so many thanks. And this is the website for my team. And we have all kinds of technical reports on there, et cetera, if you are interested.
That was awesome. I learned a lot. Thank you so much. My name is Christopher Coburn. Everybody, I use he and him pronouns. I'm not a scientist, so I won't have any graphs and data in that way. But I really enjoy getting to share the space with scientists because I always learn a lot. As I said, my name is Christopher. I'm going to spend just a quick few minutes talking more specifically about our COVID-19 response in Gallatin County. I'm going to do that through the lens of the ways in which I serve this community, which is in a few ways. I sit on the Gallatin City County Board of Health. I'll talk a little bit more about that in a moment here. I also sit on the Gallatin COVID-19 Vaccine Distribution Task Force. Talk about that in a moment here too. In my day job, I used to work at Bozeman Health, and I serve as the system manager for health improvement and partnerships. I spend a lot of time working with our community partners on efforts to improve community health. And during the COVID-19 pandemic, I had a brief stint early on in the pandemic. We're still in it, but early on in the pandemic, I served for the health system as the public information officer, which was, we probably could have used those communication strategies early on to help people understand what it is that we were talking about, what we were doing, but I served in that way as well. So before we kind of get into a few minutes, I just want to ground ourselves in the fact that the community where I get to serve, Gallatin County, is really the traditional and ancestral homelands of many peoples, including the Cheyenne, Salish, Kootenai, and Crow peoples, who continue to caretake this land as they have done for generations. And it's on this indigenous ancestral land in which we've seen this top row ring true for our experience of the COVID-19 pandemic. So in our county, in Gallatin County, we've had over 50 people who have died as a result of COVID-19 or SARS-CoV-2. We've had over 40 people who have been hospitalized. We've had over 12,000 people who've tested positive for COVID-19, and this is data as of a couple days ago. So I think it's pretty similar. It could be a little bit different now. So that's the experience that we've had in this county throughout this year-long pandemic. Our response to that has been, I think, pretty powerful, and has looked like we've locally adopted, passed, and enforced at least 10 is being really conservative because there's been a lot of changes to some of these policies, but at least 10 local health policies and rules that have been designed to keep our community, keep each other and ourselves safe. To date, we've administered just about 18,000 vaccine doses to people in our communities, and we've over 5,000 people have completed their vaccine doses, so getting those 2 shots. This is pretty comparable to what we're seeing across the state. Some other counties have more people vaccinated than we do, and that is a product of the way that the state allocates vaccine doses and how many hospitals are in each community. But we're seeing a really strong effort in Montana is really leading the nation in being able to utilize all their vaccine doses that they're getting. We have a really strong record of not wasting any vaccine doses and really getting them into people pretty quickly, and I think pretty effectively. So just to kind of talk about the response a little bit more, I want to talk a little bit about my role on the City County Board of Health, because I think we've heard a lot about the Board of Health. Actually, I know we have heard a lot about the Board of Health over the past year, and I don't know if everyone knows exactly what it is. So the way that public health operates in the state of Montana is through these decentralized boards of health and health departments. So there is a sort of state agency that Seth talked about, DPHHS, but the local decisions are made by local health departments who are overseen by and run by local county or local city county boards of health. And those boards of health are comprised of appointed people who are appointed by the appropriate commission or council body. So in our county, we have half of our board is appointed by the county commission and half is appointed by the Bozeman city commission. So that's kind of how our board is structured, and we are mandated by state code to protect the public health in a whole host of ways, including from the threat and spread of communicable diseases. So it's not something that we can do or might do. It's something that we have to do, that we shall do, and that is written in the code, which is put on there for you in case you're interested in the Montana Code annotated. So as a board, we really have a lot of responsibility. During the pandemic, it's been really focused on adopting and enacting local health rules and directives, rooted in science, rooted in evidence, and rooted in the best available information to protect people. And those have looked like restricting and closing certain types of establishments to limit the amount of time people are spending together. It's looked like enacting a mandate to wear masks when you're out in public. It's looked like instituting closing times or restrictions on who can see people in certain settings, and enforcing isolation and quarantine measures, which was like the bedrock of public health protective policy. To make those local health policies work and to kind of adopt them and to even create them, I think Seth talked a little bit about how public health is, we've learned and we continue to learn to be more effective at balancing all the different things we need to be considering that people are experiencing. So it's not just about protecting people from COVID-19. It is about that, but it's also about thinking about the other things that people need to live their lives in healthy and protective ways. So balancing all those different interests and needs that people have to create policies that are actually going to keep people safe and have a likelihood of being followed with minimal enforcement. And then we also have a big responsibility of engaging with the public. I think the Board of Health and, by extension, the Health Department have become and have been seen recently as, the leading source of information. And we really want that because it's the entire job to know the entire public health situation that we're experiencing and being able to communicate that effectively. So that's something that the board is really focused on as well. Speaking more about our vaccine distribution efforts so far, we get a lot of questions about how things are going and why they're going the way they are. And I think I want to just spend a little bit of time talking about where we're at, how we've gotten to this point of having over 5,000 people in our county fully vaccinated. So we're following an approach that's led by the state, which is grouping people into different risk phases. So phase 1A, 1B, 1C, and then 2. And this is subject to change depending on who's in the governor's mansion. And we did see that shift happen when Governor Gianforte took office. We saw a shift from the previous phases and who was in those to the current phases. And that's a challenge in public health because we are, so beholden to those policies, and we want to be in lockstep in many ways with the state and with what other counties are doing. But what that has meant and has a real implications for our county and for our state, the switching in phases has meant that teachers and other essential workers were shifted from phase 1B to phase 1C, which has a whole host of communication challenges and also just a whole bunch of community challenges when you move people around like that. But we're following the state guidance in terms of how we're allocating vaccines. To do this effectively, there is an immense amount of coordination happening between every entity that provides vaccines and every entity that really works with people or consumers or customers, which is basically everybody, because people have questions about this. So there's a lot of collaboration to make sure that we're being as effective as possible. In our county and in our state, we're have this intentional focus on being equitable as possible, knowing that the COVID-19 pandemic has been disproportionately felt by certain parts of our population, including American Indians and people of color. So making sure that we're prioritizing folks to get immunized who have shouldered the burden of this pandemic. And also being intentional about how we reach out to hard to reach population, people who don't like to use technology or can't use technology, who don't have transportation, who maybe speak languages other than English. So being really intentional about how we're doing that. And I think underpinning all this is there's really these really sort of broad and important challenges that we have that make it harder for us to be as effective as we want to be. The biggest one being supply. We simply don't have enough supply to provide vaccines to everybody who wants one right now. So the supply is by far our biggest limiting factor, followed closely by storage. I think it was Matt who talked a little bit about storage, but it is really hard for us to get vaccines out to people in non-healthcare settings if they have really sort of strict and sort of hard to achieve storage requirements. That's something that we're dealing with too. Liz talks a little bit about hesitancy, and we're seeing that as being a challenge too. It's not so much right now because we don't have enough supply, but I think as our supply catches up, we're going to have to really combat in our state these communication channels of how do we get people to move from being unsure to being excited about COVID-19 vaccinations. And then we have this situation where, in our country, it's no secret that we do not have a very well-coordinated public health system across our entire country. So trying to do a mass vaccination in this speed and to this scale is really challenging because we don't have that public health system in place like many other countries do. And the last thing I'll say is, as we look forward, as we continue to move through this pandemic, there's a few things I hope that everybody continues to do. One is this continuation of keeping a focus of keeping yourself and the people you care about in your community safe. And that looks like continuing to maintain hand hygiene, mask wearing, keeping, social distance from people, and also getting vaccinated when it's your turn to do so. Advocating for public health with your elected officials. Public health, mind-blowingly to me, is under attack at every level in the state. And so it's really important that people who care about these public health policies feel empowered to advocate for them and use their voices to say, no, we actually do want systems that are going to protect us. And then finally, I think, and probably most importantly, is continue to practice patience and kindness. We're all experiencing this pandemic very differently. There's certainly a lot of commonalities, but I found, and what we've seen throughout the response to this pandemic, is really that kindness goes a long way, and that patience sort of keep it going will really serve us well as we continue to move through this pandemic and hopefully get to an end in the not too distant future. And that